All right, let's get started. Again, welcome everybody. Last time this is the workshop session on the terahertz or T-ray, imaging and spectroscopy. We have gone through the imaging sessions, and we started the spectroscopy, particularly time domain spectroscopy. We started last time. So I'll be repeating a little bit and then go through rest of the material as much as our time permit. So as I mentioned last time, that spectroscopy is mostly about light-matter interaction, and light in the sense that everything on the electromagnetic spectrum in a broad term. So as you know, the electromagnetic spectrum has a lot of things on it, starting from radio waves all the way to gamma ray. Radio wave has the biggest wavelength, and gamma rays have the smallest wavelength. Now, X-ray, everything is on the smaller wavelength side. We focus on T-ray or terahertz radiation, as we have been doing. And the reason we do that, that first of all, as you know, T-ray were the last explored region on the electromagnetic spectrum. They used to call it terahertz gap. And in the mid-80s, Austin from Bell Labs demonstrated that terahertz can be generated. And so that got started everything. And then it came the long way. The application of T-ray is still on the rise. And, our company has found a way to generate T-ray from a new mechanism, what we call is a dendrimer dipole excitation. We have gone through that, and there's this spectroscopy section today. So as you can see in these diagrams, when light falls on a matter, many things can happen: reflection, refraction, scattering, interference, absorption, diffraction, and also light can go through, which we call transmission. Then reflection can be specular and diffused. In fact, all reflections are diffused reflection, except the specular, meaning at the very small angle incidence. Those are called the specular reflection. That has certain application. We're not gonna focus on that. Then in scattering, there is Rahman scattering, there's Rayleigh scattering, and there's mean scattering. Then absorption, there is electronic, vibrational, rotational. In fact, there is also torsional and conformational. Those things also are important. And so what T-ray has the advantage is that when you take a particular energy, for example, infrared, that is sensitive to the bond activities, bond vibration or rotation. But as a molecule, there are so many other motions or resonance in general that if we could probe, that gives us more information, more advantage. And that's the whole point of T-ray or terahertz, because it is sensitive to all the resonances that may be present in a molecule. So the absorbance follows the Beer-Lambert law. The absorbance of a beam of a collimated monochromatic radiation. Remember, monochromatic radiation is a homogeneous isotropic in a homogeneous isotropic medium is proportional to the absorption path length and also the concentration of the material. So the Beer-Lambert law, it used to be Beer's law, then Lambert modified it. Beer-Lambert law can be expressed as by this equation in which the Y is a dimensionless quantity. L is the path length in centimeter, and C is the concentration, usually expressed in mole per liter, but can be other units as well. And the epsilon is the absorptivity in case of the absorption, and expressed in liter per mole centimeter. So what happened is in a lot of cases, we can also rewrite the Beer-Lambert law. It turns out that it is not only dependent on the concentration, path length, or the absorptivity. If you keep, let's say, everything else fixed and vary one variable, for example, absorptivity, then we can replace that absorptivity by reflectance, by other material property like dielectric constant or refractive index, that kind of stuff. So it gives us a way to model and measure many physical quantities by the reflection technique. So this is what we discussed before, that we generate a pump probe technique circuit with T-ray or terahertz radiation. So the pump probe technique, as explained on this cartoon on the left side, is, there's a pump beam and there's a probe beam, and they are allowed to interfere in the time domain. That's the difference. It's not a regular interference. So when they interfere in the time domain, you get the time evolution of the interaction of the pump and probe beam that allows us to study a lot of material, delicate, undelicate, and all different kind of things. The difference is that it's in the low frequency region, because it's terahertz, from 1 terahertz to some 30 terahertz, that falls, if you convert that into wave number, it's between 1 wave number and about 1,000 or 1,100 wave number. So this is the region that is not accessible by any other radiation because of their energy or wavelength. So that is the advantage, because as you can imagine, a molecule is a heavier than the electron itself. Electron is very light compared to a whole molecule. So when the molecule has resonances, that falls in the low frequency region. And that is why this low frequency region is so important. And we discussed about this sensitivity requirement, because if you look at the even simple water molecule, that is expected to have so many absorbance states. But if you see a simple experiment like an FTIR measurement system, they're taking 3 different fruits, trying to understand how they absorb light, and as you can see, you get a single peak within that wavelength because of the sensitivity issue. I mean, in any of these fruits, there are a lot of water molecule as well. So you expect to see a lot of absorbance peaks, except the sensitivity of the, it's a combination, not only the radiation, but also the instrument. If the sensitivities are there and instrument is not able to record that, then we're not going to see it. So both the radiation, which is exciting the higher frequency, which is, you see, 500, 600 nanometer wavelength is in, multi-hundred terahertz frequency. Therefore, the frequency is one factor, but also the instrument also has to have the ability to sense whatever is there. And then, we talked about the diagram. I showed just here, this one. This is the spectrometer. Then the spectrometer needs calibration, and calibration has been done with respect to water molecule, with respect to polyethylene molecule. I'm skipping because we have discussed this before. And then what happens is the interferogram, as we generate in the time domain, need to be converted to frequency domain by Fourier transform. But the point I'm trying to make here is that a Fourier transform is not a single mathematical relation, which, Joseph Fourier, one of the greatest mathematician who first formulated this Fourier transform that converts the time domain to frequency domain, any variable, or vice versa, inverse Fourier transform. We'll do the other way around. But then, subsequently, a lot of other mathematicians and scientists have contributed and came up with different kind of transformation capabilities. Because in one situation, one transform is very good. At another situation, that transformation may or may not be equally effective. So the software that's bundled in the system has this regular Fourier transform, which is per the original Fourier transform. Then there is Fourier transform with data window. I will explain what all of this means with examples, but so that you know that all different kind of transformations are there that you can convert the interferogram, which is the time domain evolution, or the interaction between the pump and probe signals in the time domain with the this whole suite of tools that are useful in different situations. So in other words, it's not a one-trick pony, so to speak, right? You can have all different kind of advantages. So again, I showed this before, but these are the other work that was compared for calibration. We discussed this before. So I'm gonna skip. This is polyethylene, interferogram and how the peaks come up. We have compared them with the published ones, the ones that are published in these references, and the ones that T-Ray has measured. So some of them match very well, but there are other peaks that were not reported before. What do they mean? We still have to figure that out. This is the job for some astute scientists to sit down and figure out why we see a peak at this wave number. Is this real one? If it is a real one, what does it mean? The tool gives us the opportunity to discover this kind of things. And we also went through this endohedral system, which is the hydrogen molecule encaged in a buckyball or C60. And then the restricted motion. Very convenient way to study both the buckyball as well as the endohedral system. And we know that this buckyball alone has so many different kind of resonances, vibrational states in there. There are more. I'm giving you just these three examples, but there are quite a few that are available from this reference, which is the work produced at the Arizona State University by Smith. Was there by Smith, and I have the reference somewhere. Menendez and Page. So sorry, not Smith. Now, so these are the states that they have reported. This Menendez and Page at Arizona State University. And we have seen that a lot of them we can match, but some of them are not reported. And some of them reported we couldn't match. So always. Here is an example of FTIR spectrum of the C60 and H2 at C60. H2 at C60 means that hydrogen is encaged inside the C60 molecule. And that's a famous paper came out from Japanese group in encapsulation of molecular hydrogen in fluorine, published in Science, and in 2005. So FTIR couldn't discern between the C60 alone and when there is hydrogen inside the C60. And the reason being that if the hydrogen is inside the C60, you need a radiation or energy that could penetrate the carbon or C60 molecule cage and interrogate the endohedral molecule, hydrogen, in this case. So that's one of the advantage that T-ray offers us, because when we take interferogram with terahertz, spectrometer or time domain spectrometer, so this is 6 picosecond on the x-axis, and so we have a C60, which is pure C60, and then endohedral with hydrogen and endohedral with the deuterium. So this gives us a very nice way of comparing what happens to C60 when you put impregnate it with hydrogen and deuterium. So what we have seen comparing to the work of Menendez and Page, we see that a lot of the things that comes close match, but some of the things are slightly different. So experimental condition, a lot of these predictions have been done theoretically. Some of them are experimentally found. So quite a bit of sorting out and research can be done with these molecules. The motivation and why would one do that is the fact that there is ortho and para. So if they engage ortho species in the C60 and para species in the C60, they give totally different signal. And that has implication in the NMR spectroscopy. That was the main motivation of this work. Another example here I'm bringing is non-ionic detergents. These are called tritol and triton. And so this one is triton and this one is tritol. These are the non-ionic detergents used in biopharmaceutical manufacturing. They, particularly cancer vaccine and that kind of stuff that has been the strong focus of National Cancer Institute, along with a company called Leidos Biomedical. So we had a chance to work with them a long time ago. Before they were Leidos Biomedical, they used to call SAIC Frederick, and that's who we worked with. They provided this X100 and NP9, which is the generic name. But you see, biggest difference is that you have 8 carbon and you have 9 carbon. So because of this, closely spaced molecular structure, FTIR practically produces the same spectrum for both of these. So how do you know which is what? And that's where we do terahertz. When you do the T-ray spectroscopy, the carbon 9, which is the blue spectrum, has higher loss or absorbance. Is in dB. So decibel is the unit of loss, right? So higher loss compared to the triton, and that makes sense. And all of these peaks can serve as the identifying factor or signature for this entire molecule. You see that in the lower frequency range, there's a strong signal here at around 5 terahertz, and the triton doesn't have that. Triton is a little lighter molecule, therefore it shows a strong peak around 8 or so terahertz in this area. So this could be the signature for identifying these molecules, and we have other examples of getting into that. I want to take a moment here to reflect. If anyone have any question or comment or point, you can make it now up until here. So moving ahead, this is another work. What we have done is this is also a collaboration with Hershey College of Medicine, and they are able to synthesize the DNA molecule of certain length. So there's a lot of work. I'm showing here the spectrum, but there are a lot of work that was done in this area. Particularly, we were interested to identify or learn the signature of what they call as a single nucleotide polymorphism. And I did not introduce a lot about that, nor am an expert of that by any chance. But the point is, human DNA, a huge, long molecule, right? In which there are segments that are known as gene. And this gene is the one that determines a lot of things in human body, including the fact that, let's say somebody can drink 10 cups of coffee a day and still get sound sleep at night. And if I drink more than 1 cup, I'll be up whole night. This distinction comes from a single base pair difference in the gene of that particular gene in these people. Same thing, more important is that same medication, scientists are always coming up with new medication. Let's say there's a medication that works very well for some disease on some people. The same medication may or may not work equally well on another person having this kind of discrepancy in their gene. That gene that is, responsible for that kind of activity. So, and I have a full-blown article on it as a newsletter. If you are interested, I'm happy to share with you. But there are, 10% population has more than 10% population has this kind of genetic variation. What they call is a single nucleotide polymorphism. Nucleotide is also known as base pair, also called oligonucleotide. So we call oligos for shortcut for oligonucleotide. Sometimes they are called base pair because they come in pairs. The double helix of DNA molecule, they're always in pair. And what happens in that gene that one of the base pair got changed. One of the 4 constituent of the DNA, which is ATGC, adenine, thymine, G is guanine, and C, I forgot what C is called. So this 4 thing, makes our gene. Each of them is called a base pair, also called a oligonucleotide. So when in a gene, let's say, supposedly this is 559T is an identified gene, but we have only some 21 MAR here synthesized. And T, because it has T, and instead of T, they're calling it G, the same number, but G because in case of T, there is G here. So this is G mutation, for example. And so in the paper that we have seen that how is the absorption of individual oligos or oligonucleotides. And we have seen that, and I would love to share that paper with you, but I even don't have any reference here, do I? No. So what happens is we have seen that G has, so we took 4 samples of single species. In one species, all A, in another species, all G, all C, and all T. That's the experiment we did to find out which one absorbs the most. And we found that G absorbs more than T. And how much more is all listed in that paper. I'll share them later. So again, when we did the spectroscopy of this particular strand containing a single nucleotide polymorphism with T-ray, we see that G has a higher absorbance compared to T. And also, so this distinction, again, if we did this with FTIR, would not be able to be visible. So that sensitivity is provided by the T-ray, as well as, of course, the instrument is able to measure it or record it. And then the same strands was hybridized, meaning that you can have DNA as a single strand, which is not a DNA yet, and then you can hybridize them or make double strand or helix. That's called hybridization, also called double strand. These are the biochemists and biologists. They are expert in those things. We got the samples again by collaboration with Penn State College of Medicine in Hershey. And so question became that, okay, if you see the difference between the single strand for single nucleotide polymorphism, when they are hybridized, can you still identify them or discern them? And that is the experiment we saw here. So this red curve is only A, adenine, and the blue curve is only G, which is, the transmutation from T to G. And then the A, this A plus C, B plus C, these are the strands. So A is this strand here, B is that strand here, and then A plus C, meaning that A strand is hybridized with C strand, and B plus C, meaning that B strand was hybridized with C strand. And if we did that, then this green curve is A plus C compared to A, this red curve. And then blue curve is B, but B plus C is this brown curve. As you can see, when you hybridize, because it's much heavier now and different constituents are there, so the signature is quite different than the single-stranded ones, which makes sense, which is expected, as well as now we can figure out if we could, the key would be that if we can reproduce this experiment over and over again and learn what these peaks mean, then we can use them as a signature. In fact, there is a possibility that one can do the genome sequencing without PCR machine. But we haven't done anything close to that. But there's a possibility that this technique could be used for genome sequencing. Okay, now I'm switching topic again. Spectroscopy. Any questions up until now or comment? So spectroscopy gives us a lot of application opportunity. This was also a collaboration with the university, Shanghai Ocean University. This faculty member who graduated from some US university, went to teaching over there, and question was that there are a lot of produce, meaning vegetables and fruits. They're growing, farmers are growing them and using a lot of pesticides. So is there a way to detect the pesticides as we go and purchase them, bring home, wash them, eat them, but is there any pesticide within that produce at all that we should be mindful of? That was the question. So what was done here is this carbaryl is actually a molecule present in some pesticide. So we had that carbaryl, and we had a very smart intern who did all of this work. So what we did is we sliced the apple. This is an apple slice. And we had one apple slice that is untreated. The other apple slices, separate apple slices, were treated 4 different concentration of carbaryl. Or 2, I don't know. I think 2 different slices, concentration of carbaryl. In one case, it is only 5.1 ppm. In another case, 13.9 ppm. You don't want to go too high a concentration because you wanna get into the region where practically it may happen in the produce. So this is a spectrum over a long frequency range, but nothing we can learn much except for their loss factor. The untreated one has less loss. Higher the concentration, higher the loss, which makes sense, but that doesn't give us a way to find out whether this particular pesticide is in there or not. So what was done, you see, remember a few slides ago I showed there are so many ways of doing the transform. So one of them is what they call is a eigenanalysis frequency estimation. So this eigenanalysis becomes very handy when we want to see here we have, all little things, do they mean anything or not? We have no way of knowing. Probably they don't. But when you boil them down to the eigenfrequency ones, then you can see that the untreated one has this particular peak. Then the treated one with the small amount of carbaryl and then a little more amount of carbaryl. And you see there are, in this table we're showing that the untreated apple has a significant peak or a discernible peak at 2.99 terahertz. When you add carbaryl to it, you still get those peaks because the apple is not changing, apple is still apple. But what happens is these 2 peak, pure apple had this 12.52 terahertz and 21.43 terahertz. Instead of that, when you add this carbaryl, it shifts to, instead of 12.52, it turns to 13.96. And instead of 21.43, it goes down to 16.06. So this kind of shift, and they're not changing because of the carbaryl concentration. It is the specific to carbaryl chemical. Therefore, you get at the same region. And that can be a signature of the presence or absence. If we see these peaks, then we can say, okay, carbaryl is here. If we don't see those peaks, it is not there. So that's another way of utilizing this spectroscopy. So that's, I think I'm switching the topic and maybe we can stop here today and get to the other topics some other thing next time when we can open for discussion or question, comment, etc.
I have a question.
Yes, please.
If you can go back to that slide of carbonyl peak in treated apples.
Right, sure.
And you said the carbonyl peak is 13.9.
It is not carbonyl, by the way. It is carbaryl, which is a different chemical. Pesticide.
Oh, okay.
In pesticide, yeah.
Right. Two parts
I'm
in
sure.
my question.
Yeah.
Two parts in my question. First one is, these 2 samples are treated in the laboratory with that concentration, right? Not out of the garden. Not out of the garden.
Yeah, this apple was sliced in the lab.
Right.
And then carbaryl was added to it. Yeah.
Okay. Now, for some cases, the peaks are around 13.9 or, with some variation between the two concentration.
Yeah.
But for the higher concentration, the shifts are very high, like 16, around 16.
Yeah.
And is it standard for that peak?
No, it is not standard. It happened that way. So, because higher concentration makes it heavier, so it could come down. The key here is to, if we find the same peak over and over again or not. That's the key. So the question is why compared to the the Apple alone had a peak at 21.43 terahertz, and why we are picking this 16.06 or 16.19 or around this here, because the reason we're doing that, this is reproducible. This is repetitively coming into the spectrum, meaning that even in higher frequency range, there are some other peaks, outside 21.43, but those are not very prominent. And that is the reason we chose not to entertain them. But this 16 is there. As soon as we put carbaryl in there, the 16 point some peak comes in. So that constitutes a signature for that particular chemical, or carbaryl in this case. That was the rationale. Again, a lot more work could be done in this as well. The intern, when she left, we didn't do much about it anyway.
Right. Another question related to that.
Yeah.
Just to a little more in terms of treating the samples in the lab, and those two concentrations. So are those treated in a specific way for same amount of time or variation of time?
Yeah, I think what was done is, so first of all, we found a slicer, so they're more or less identical slices are done of the apple. And then the carbaryl was dispensed, and the solvent was dried at room temperature. And so it was not heated or anything. And so there is a particular area where the carbaryl solution was applied. And there must be some source of error there, but within the experimental error limit, this is what we were able to do.
Right. Understood. Now, final question is, for the original peak for the untreated apple, 12.52 and 21.43, those two peaks still there for the treated sample, or they disappear?
They have shifted. For example, the 2.99, this peak is in all samples, treated or untreated. And so this seems like coming from the apple itself.
Okay.
But other peaks in the 5.91 in the untreated apple compared to 9.53, but the higher concentration brought it down to 6.86. Seems like it is due to the carbaryl.
Okay.
So it's, I don't remember all the details right now, but we can always go back and find out what was done. But these two peaks appeared over and over again in repeated experiments. That's the reason we focused on it. I don't know what happened to this. This might have there or not. I'm not sure at this point.
So it's an obvious thing, but I am assuming that the carbaryl peak samples were also done by the pure sample, pure non-apple sample with the chemical only for matching purpose.
Oh, No, that I don't think we did that. The coverall alone, I don't know. Oh,
Probably done more details, but something to look at.
yeah, it could be done. Yeah, could be done. That is a great point because if coverall alone produces a signature, and then that would be a true positive when we see it on Apple as well. Yeah, very good point. Sure.
Another question, then I will stop. That's going back to your Beard-Lambert law measurement for three fruits.
For this one? No, sorry.
No, the absorbance with three fruit samples.
Oh, okay. Sure. Maybe I should find that paper and share that. This one?
No, this is your DNA stuff. Earlier.
Okay.
Earlier, I think. Somewhere.
This one?
No.
Okay.
I don't remember number of the slide, but
no, that's okay. What is the topic again on that slide? What sample?
The topics were the three samples for Beard-Lambert absorbance. The three fruits had different colors, so absorbance were different.
I believe this one.
These are fullerene. These are not fruits.
Fullerene. Polyethylene?
No, not yet.
And so we started today. Yeah.
Go slowly down. Anyway, yeah, up. I say it up a little bit up. No, further up.
This?
Yeah, here, I think so.
Oh, okay, yeah.
So question is how the samples were prepared, because these are the samples from the fruits. At the end, before the measurement, the samples were transparent without any suspended particles?
I have no idea. This is from a textbook. So we just
okay, I see.
Borrowed.
Never mind.
I was teaching a course on this thing, so I had to borrow from that book.
Okay.
So I'm not absolutely sure. What they're saying is that they have UV light source, visible light source, deuterium arc lamp,
right,
incandescent bulb, all different kind of things they try to compare. So basically, this is a commercially available spectrometer.
Sure. I got it.
UVBs, yeah.
Okay.
Yeah.
No, never mind. Thank you.
Oh, this is a very good discussion. I guess, learning is always a give and take process. So a lot of things comes up. Unfortunately, Miles is not able to talk today, I suppose, because he's in a library.
That's right.
Farin, you want to make any point or anything?
No, I had some questions about that. Dr. Mujumdar cleared a lot of stuff, so yeah.
Okay. Wonderful. So then I think we should be able to get done in the next or so lecture. As a heads up, what we want to go through is this thickness profiling. This has to do with the transdermal drug delivery and early detection of skin cancer. So those are the things we will do, as well as a few other things. Probably next 2 sessions should be enough. And by this time, I hope Miles and Farin have some topic, and Dr. Mujumdar may also want to give us some outlook, but then we can get to the closure of the workshop, and we will have a certificate of participation, and hopefully that will be, we want to finish by the end of this year for sure, hopefully by November.
Sounds good to me.
Okay, so
And by the way, I apologize for not being able to join.
no,
for
you
almost 5 months.
you had to do what you had. I know that that's not easy. So let's stop recording.